Zoe. It's a pleasure to be here. I hope next time we can be in person. So I'll just start the slides. As Zoe mentioned, yes, I've been involved with FEP calculations for a long time. I'll show you a little history of that. But what I want to share mostly today is our discovery of inhibitors of the main protease of the SARS-CoV-2 virus. We've done this extremely quickly, and it was absolutely essential in our rapid advance to be using FEP calculations. So, for the community, this is a real success, demonstrating the immediate importance of doing FEP calculations. So here's a group. We do have support from NIH, also the Yale School of Medicine gave us a little support to begin the work on the coronavirus. But I'll go back to the history first. So just particularly maybe for some of the younger people, this is our free energy journey. And basically before 1980, there were virtually no free energy calculations going on involving simulations, because there wasn't enough computer time to do much in the way of modeling of any type of fluid system. So they really started in the late 70s and in the 80s. Again, the tip 3P and tip 4P water models weren't developed till 1983. The water models before that were awkward, and they didn't give good properties of liquid water, in particular the density, because people didn't do NPT simulations. So it was very, early days. Then a number of us did get involved in free energy calculations, and the method of choice before 85 was umbrella sampling. And so I won't go into detail on that, but we did this calculation that's well known on the SN2 reaction, studying solvent effects, and that was all done with, again, umbrella sampling. And then in 1985, various events led me to consider trying an FEP calculation, including I'd read the chapter in the McCrory book on what we called statistical perturbation theory, which sort of morphed the term into FEP. But I wrote the code in the BOSS program and just wanted to try a simple example of an FEP calculation. This was very exploratory because it just didn't know if it would be numerically stable, if maybe the error bars would be horrible. It was just absolutely exploratory study that turned out to be extremely favorable in terms of the precision of doing FEP calculations. But this was the first, I would say, modern FEP calculation on a molecular system, introducing things like, at that time, what we did is the Lambda modification we did in what today would be called single topology mode. But this was very exploratory, and fortunately it worked out well, and the rest is kind of history. But there were still very serious limitations in computer resources. So people weren't doing calculations on proteins in water significantly before about 87 or so, and didn't become at all common. This is, BMD, or we were doing Monte Carlo proteins in water were, again, very uncommon. Then, skipping ahead, by 1997, though, we were doing FEP calculations for protein ligand systems, but it was still early days for that type of study, again, because of the computer resources that were required. But then we did gain quite a bit of confidence in our ability to get reasonable FEP results. So by 2005, we were doing FEP guided drug lead optimization. This was always been a goal, is can you use FEP to design inhibitors? And we've worked over the years and shown the viability of this, particularly with our work on developing anti-HIV agents. And then subsequently, Zoe, for example, worked on our MIF project. and we've worked on kinases. And this all set the stage for us to tackle the main protease of the coronavirus in 2020 when this urgent problem came about. So just again, briefly, I think you know this. This is the Zwanzig equation from the 1954 paper of Bob Zwanzig. He was a postdoc of Kirkwood at Yale at the time. And then our reference for our 1985 paper. So Zwanzig paper, the FEP there was from a rigid sphere to a Leonard Jones gas particle. And then ours was this ethane to methane, methanol perturbation in tip 4P water, 1985. But once that is established that you can do this type of alchemical transformation, everything else just falls out. You can do various free energies of transfer. So protein ligand binding is just a free energy of transfer calculation between unbound in water and bound in the protein. And then of course in the QMMM area, there's a lot of interest and utility in doing free energy profiles by perturbing along a reaction coordinate. We continue to do methodological studies. Here are a couple of papers from 2019. Those are the original references. There's the Zwanzig paper from JChem Phys in 1954, and then our 1985 JChem Phys paper, and the curve that we got for the methanol to ethane calculation going forwards and backwards, and having this very high precision. Again, accuracy is, yes, we agreed with experiment, but the precision, which was key here, was that there was very little hysteresis in the calculation. So we had an error bar of less than 0.2 of a kcal per mole in the free energy change. And that was just, at the time, to me, stunning, unexpected, and it really then caused a lot of excitement with other people trying it, though at the time, these calculations were thought to be extremely expensive. Like this calculation took about 6 weeks on our one computer. Okay? We had one CPU available to us. All right, so let's go on. I think you know a little bit about coronavirus too. And one of the key molecular targets is the main protease. The main protease, if you inhibit that, then the polypeptide that is created from the translation of the genomic DNA is not cleaved, and then the virus can no longer reproduce. So it's a very good target. It's also a good target because it doesn't mutate much. There's 96% sequence homology between the SARS-CoV-1 and the SARS-CoV-2, and the differences aren't even near the active site. Whereas the spike protein is is the troublemaker because it is mutating a lot. So we're immediately looking for inhibitors of the main protease, and we have this substantial history of working with viruses, namely HIV. And this work, the design and synthesis is done in my laboratory. And then we work with our collaborator in the med school, Karen Anderson, and her people on the assaying. And so the thought I had while we were locked out of our lab from March until June of 2020 was to see if we could find some starting points so we can do standard docking, and we've done that. And then I also felt that we should try docking known drugs, and I keep a library of 2,000 known drugs. That's about all the oral drugs there have ever been. And I asked Mohammed and Julian to dock it, and they used 4 different docking protocols, so consensus docking, and processed this, and we got some compounds that are then hand-picked that we were going to purchase an assay. So I ended up hand-picking 17 compounds that I thought looked like they might show activity. Plus, these are things that I hoped that in our lab we could analog. So I was looking for compounds that we might be able to make analogs of to improve the potency. I didn't expect to be successful at repurposing. Repurposing is virtually never successful. But I was hoping to get some starting points. And fortunately, that's what happened. So you can read this paper. Of the 17 compounds, 14 of them showed activity in a standard inhibition of proteolytic activity assay, where you use a FRET peptide, and when the peptide is cleaved, you have a fluorescent EDANS group that you can use to monitor your success at inhibition. And so here are the compounds that we purchased and then assayed, and this is just the inhibition of the activity. So this minidipine is the best one. And then we did determine IC50s, so the activity 100 micromolar concentration is shown here. And then we did determine IC50s, and the best ones were in single digit micromolar, which I thought was terrific. And then we got, of course, from the docking, we had pictures of all of the predicted complexes. Here's the binding site, and it has a number of features that are referred to as the S1 pocket, S1 prime, S2, and then this channel, S3, S4, S5. And here's just a compound, azolastine, that's docked into that. And this is one I looked at and felt, we could probably analog if we had to. And so that was a possibility. So we published that paper in MedChem Letters. So the next step was to pick a compound and then do lead optimization guided by FEP. So here is, in fact, the compound that we picked, I picked, called parampentyl. So I didn't mention anything about parampentyl in this paper, but I was thinking about analoging. It wouldn't have been obvious, I think, to the average reader that we were going to pick parampentyl, because it wasn't a very good inhibitor. It was above 100 micromolar in the assay. But I felt the structure was compelling. This is the docked structure here. Here it is. I'm lining everything up in exactly the same way. First of all, this is a triaryl something on a core. And that's the type of thing you can make relatively easily by coupling chemistry, like Suzuki chemistry. So I felt we could work with this. I also liked the docked pose. It looked very reasonable to me. And I could immediately see features that were not optimal. So for example, this carbonyl group wasn't hydrogen bonding to anything. There's an NH over here that wasn't hydrogen bonding. This pyridine ring of parampentyl the nitrogen wasn't doing any good. This phenyl ring over here is in a fairly polar area, so that's a mismatch. So by eyeball, I could see a variety of problems with parampentyl. And then what I did is FEP calculations, and these are things I actually do myself. I have other people like Mohammed and Julian helping. But I ran all the FEP calculations I'll show you here by myself. So the first thing I did, and I'm showing actually my raw data here from my Excel file. So here's the parampentyl, except I've taken that phenyl ring now, and like I said, I didn't like it, so I wanted to change it into a heterocycle that would match better. So I said phenyl to 2-pyridyl, 3-pyridyl, 4-pyridyl, 4-pyrimidine, triazine, and an N oxide. So I'm trying different things here. This is the ring there. And here's the delta G's I got. So I have the bound and the unbound, and only one of them looked any good, better than benzene, and that was 3-pyridyl. This right here. And the reason it looked good is it forms a hydrogen bond with the NH of this histidine. So that was obvious then. We had to do 3-pyridyl at that for the aryl group. The other thing was pretty clear to me, this carbonyl in parampentyl, again, as I said, wasn't doing any good. But if I moved it over here, Zoe, just checking, is my pointer visible when I?
Yes, we can see it.
Okay, good. If I moved it over here, I might be able to pick up the hydrogen bond with that NH, or the backbone is glue 166. And so I did an FEP calculation on that. So moving the carbonyl from what I call the 2 position to the 6 position, and gangbusters, that was really favorable. So that's a winner. And then the other thing is I knew that this group over here could be optimized better. It had the nitrogen, like I said, in the wrong place. So I just did a chlorine scan on that, and that gave a very clear signal. So full chlorine scan, or through the all possibilities, that what I wanted was a metachlorine. And I had eyeballed that previously, but this confirmed it. But these FEP calculations were very molecule. So here's parenteral. Recognize that I could probably replace this phenyl edge here with a uracil and pick up some hydrogen bonds with a threonine 26. So that was by eyeball. And then, of course, if you have the monochloro, we knew we could put in another metachloro here and you gain a factor of 2 from the symmetry benefit. So now you just have to make the compounds, which I turned to Chun Wei in my lab, and I borrowed Lizzie Stone from Scott Miller's lab, and they started making compounds. And this is what happened. So these initial designs from FEP and a little bit of eyeball, we went from more than 100 micromolar to 10 micromolar, 6 micromolar, and 4 micromolar. So we're rolling, and thank you FEP for giving us confidence. The software I'm using here is MC Pro, and the force field is OPS AAM for the protein and OPS CM1A. So I'll just take a 1 slide break here and mention the OPS CM1A is available for this LigParGen web server that I think many of you are familiar with. And so you can just enter a molecule any way you want, an organic molecule, and you'll get out a parameter and topology files for running most common software. This is being pretty heavily used. In the last 30 days, 6,000 visits to our server. So please continue to use the server, and we're happy to have provided this service to the community. All right, so now we have these single-digit micromolar compounds. We'd like to get a crystal structure, of course, which we did with in conjunction with Karen Anderson again, our collaborator. And here it is for the compound with the cyanophenyl, and it's exactly as we predicted. So this is absolutely identical to what we had modeled. There's the hydrogen bond with the backbone of glue 166, the hydrogen bond with histine 163, the cyano group's forming a hydrogen bond with the NH of cis 145. So now we're rolling. We didn't get into the lab until June of 2020, so there was no lab work done before then. By October, we had, as I'll show you, 20 nanomolar compounds. So next thing is we decided I wanted to grow into this S3, S4 channel. So I could see there was room here above this chlorine. And so what do? Well, you do some FEP, and I built structures with my bond program, but then the ones I think look nice, we're going to do some FEP. So I changed that chlorine into methyl ethyl propyl, methyl ethyl propyl, butyl, etc. And then I knew these weren't going to fare well because you're always taking a polar substituent. Just it's at best a wash when you have a hydrogen bonding group like that. So, but anyhow, hydroxymethyl, hydroxyethyl, and also just a mom group. And this very clearly said we should be putting O-propyl at that position, or maybe O-butyl. This methoxy, ethoxy would be okay, but that's what we wanted. And so we next then synthesized this O-propyl compound in both the cyanophenyl and the uracil series. And trifluoromethyl is also likely to be good. The general worry you have with trifluoromethyl is you may start having solubility problems. But trifluoromethyl is more hydrophobic. It's going to fit into this pocket we'll see in a second that we call the S4 pocket. So we made those compounds, and we're doing real well. We got 140 nanomolar, thank you, FEP, 120 nanomolar in the uracil series. We go to the trifluoropropoxy group here, we're at 72 micromolar, 72 nanomolar, and in the uracil series, we're 25 nanomolar. Now, I have to say, this compound we synthesized a little later here, but that's a 25 nanomolar compound. So again, thank you, FEP. We got a crystal structure for these compounds. We probably have 15 or so crystal structures. Here it is with the cyanophenyl series, and there is that propoxy group. And we had predicted by our bomb and calculations, it would have a gauche terminal dihedral angle, and that was confirmed by the X-ray. And it's exactly, again, as we predicted. And this is the S4 site where that terminal methyl is sitting, right in that S4 site, which is above this methionine and below this leucine. So what do we do now? Well, by bomb, I'm also modeling other possibilities here. So O-aryl type possibilities. And the benzyl oxy looked good. And so just from bomb, we wanted to make this benzyl oxy compound. And then whenever I see a benzene ring, I'm going to do a chlorine scan on it. So we did a chlorine scan on that, and it said, here are the results. We also did a methyl scan, and it said put in a methyl group ortho or a chlorine. Those are going to be the winners. And so here we made the parent compound, but then I made both the methyl and the chloro ortho analogs. And what happened? Well, the parent is 128 nanomolar, and sure enough, that orthochlorine ends up sticking, as bomb predicted, right into that pocket, and that's an 18 nanomolar compound. Okay, so again, very fine prediction by FEP. And then we have a crystal structure of this compound I'm calling 14. And there's the uracil piece showing the 3 hydrogen bonds that Baum had predicted. And I had initially eyeballed the other hydrogen bond to the carbonyl hydrogen bond. So this is a very nicely hydrogen bond compound. And there's the benzyl terminal rings sitting smack where we want it to be in the S4 site. All right, so then what do? Well, we published the paper. So this is now published, appeared, and so we submitted it by the end of January. It's published the end of February in ACS Central Science. And in that paper, we published the compounds I've been showing you. We published 27 compounds in all, starting with parenteral, bringing us to quite a few compounds that are in the 20 to 40 nanomolar region. So this is now inhibiting the enzymatic activity. To be serious here, we're going to have to do cell assays, which I'll come to next. Okay, but we have very potent enzyme inhibitors. Other people have struggled, to get anywhere near where we are. So these are very, very unusually potent compounds that we got to very quickly with the guidance from FEP. As of June here, we've made 65 compounds. We have, like I said, about 15 crystal structures. We now have just submitted a paper recently where we report further optimization of this S4 site, introducing 5-ring heterocycles. So the compounds look like this. We've also methylated the uracil, which turns out to be important for the cell activity. 8 of these new compounds are in the low nanomolar range. Now to the trouble, some aspect of working with the coronavirus. When you're doing the cell assays, if you're using cells infected by the live coronavirus, you have to work in a BL3 facility with additional breathing apparatus, because this, of course, is airborne. So here's our expert, Krasimir Spasoff, who works in Karen Anderson's lab, suiting up to work in the BL3 lab. So in the recent paper, in the first paper, we reported some of the cell assay data, and now I'm reporting additional data for the compounds in the second paper. And here it is. So remdesivir, I think you've heard about. It's a nucleoside. It doesn't inhibit the MPRO, but it shows antiviral activity. This is the IC50 in the enzyme assay. Now this is an EC50. This is a cell assay. These are Vero E6 cells. And here they are infected with live SARS-CoV-2. So this is, nasty business. And we have assayed remdesivir ourselves with the infectious virus that gives us 2 micromolar. This 0.77 is for literature value with a little bit different conditions. These are some of our compounds. So we have a bunch of compounds that are as potent as remdesivir. But the very good news here is we have a compound, number 19, shown over here, which is 80 nanomolar. So it's an order of magnitude more potent than remdesivir. We also have what's called a replicon assay. Replicon, you have a non-infectious form of the coronavirus. So you replace the spike protein with an alternative. So this is an assay you don't requires the BL3 facility, but it parallels very much the assay using the infectious virus. And in that assay, we also have compounds that are looking very good, 500 nanomolar or 175. But this compound 19 looks really dynamite. Here are CC50s, and these are lung cells, and then the Vero cells. And we have very little cytotoxicity. The compound 19 is very soluble in water, which is good, but its solubility is a little bit limited in DMSO, which limits the dilution series here. So this compound, as far as we're concerned, it's not showing any toxicity. So compound 19 is quite compelling. It's 10 times more potent than remdesivir, shows no cytotoxicity, excellent aqueous solubility, 117 micrograms per mil. And we've also sent it through PAN labs. It has no inhibition of cytochrome P450s and no significant off-target activity. So that's basically where we are. I hope you can all be happy that FEP has helped to drive this project forward, and we are trying to further develop this compound series with a partner. And here's, I don't think this crew is that interested in synthesis, but these compounds aren't that difficult to put together. You have a couple of coupling reactions. So here's a Suzuki reaction, and then this type of reaction where you bond to an NH, such as this is a so-called Chan-Lamb coupling, and then followed by another Suzuki to put in the third aryl group. Other people, what are they up to? Well, of course, a lot of people have been doing covalent inhibitors. Here's a Pfizer covalent inhibitor, JMedChem. This is on SARS-CoV-1. It's only 4.8 micromolar EC50 in the cell assay. This one over here from Nature Communications, the best compound pair, 4 micromolar. Again, we're 80 nanomolar, that compound 19. this paper in 2020, 720 nanomolar. The COVID moonshot project, there's a lot of effort gone in there, 1,700 compounds. Their best IC50 is 80 to 100 nanomolar. Here's some more. So just to give you a sense that our compounds here are quite unusual in how potent they are. And again, they're non-covalent, they're non-peptidic. So that's where I'll leave you. I just want to highlight the people in my lab, and you could also see in the paper, we have a lot of co-authors because of all the assay work. And then the two key synthetic people, though, Chun-Wei Zhang in my labs made all the uracils, and Lizzie Stone from the Miller lab made all of the cyanophenyl compounds. I always have to thank my senior associates, Julian, senior research scientists in the lab, and Anna and Joe, who are associate research scientists. And so I thank you very much for listening, and I'll stop there.
Thank you, Bill. That was really exciting. I mean, you walked us through from 100 micromolar, I believe, to 18 nanomolar. There's over 5,000 increase in potency. That's fantastic.
Yeah, and that was done between mid-June and mid-October with two synthetic chemists.
So
you
just
can see how few compounds we made to let that be possible.
Wow. So that's really showing us the power of a prospective drug discovery of FEP. That's fantastic. So let's take one question because we're 15 minutes late, and then we can go to the poster session. There's a question from Callum Dixon, who asks whether FEP often overpredicts. For example, a six-fold 120 nanomolar versus 20 nanomolar was predicted at -3 kilocalories per mole. And if this is a trend that you typically see, that FEP overpredicts.
Yeah.
And whether do you care as long as FEP predicts the best compound you can make?
Yeah. Okay. So it's a very good question, and it's come up before. As a medicinal chemist, all you want to do is know the right qualitative answer, and then you'll go forward and make the compound. So if you can predict, what is going to be more potent, whether you get a factor of 2 or a factor of 10, you're happy with any increase in potency. So it doesn't have to be perfect, but you want it to be as qualitatively correct. Yeah, our numbers, we've discussed this before, our range is always too big compared to experiment. And we attribute that to two things. There's not enough protein, we don't pay the penalty sufficiently for protein relaxation on the binding in the way we do the FEP calculations. So I think it requires a lot more sampling flexibility. And then point B I've also made is these force fields, particularly when you're developing your charges to give correct free energies of hydration, are overpolarized. So they're too polar in the protein. So things like the hydrogen bonds with the protein are too strong. So you end up having the interactions with the protein too attractive with these fixed charge force fields. So we understand all that, but like I said, if you can get the right qualitative answer, you're golden for doing drug design.
Thank you so much. There's a couple of other questions in the Q&A box. If you see on the main screen,
okay.
There's a few questions, and if you want to answer them, you can just hit on the blue button, type answer, and then just answer them in writing.
All right. Well, thanks, Zoe.
So thank you very much. Here's the virtual applause. So it was a fantastic talk, and it was an honor to have you with us.
Okay, appreciate it. All right, I'll answer. Okay,
great,
bye.
thanks. Bye-bye. And for everybody else, we can move on to the poster session. I just typed in a link on the chat box where you can click on the link, go in a new Zoom session, and follow our poster session. So with that, I'd like to thank also Jonah and Bill for this wonderful session. And we'll see you all back here in this room after the poster session finishes. Thanks.